This is kind of a general presentation concerning the hypotheses which underlie the effects of the deuterium depletion. I will not present data. Most of my data can be found in various publications. And I will rely on several other documents that have been published on the same subject. So please, the ones who are physicists, bear with me. I am trying to present a few general things. So the water is the most common compound on Earth, but its composition is not absolutely identical throughout the world. The most significant difference is represented by the inhomogeneous isotopic composition due to the so-called kinetic isotope fractionation. So it means in fact that whenever water evaporates, it's going to lose a few of its heavier molecules. So the more water is obtained, the more drinking water is obtained from condensed water, the lower is going to be the concentration of deuterium. And you have already discussed about the Greenland ice and the mountain ice or the mountain waters and so on, and perhaps some of the benefit of living in the higher altitudes and in the mountains might be, let's say, connected to a slightly reduced concentration of deuterium in the drinking water, and of course in all the other food elements harvested throughout the area. This phenomenon is due to the fact that the heavier molecules, like in the case of water, the D2O or H2O, will accumulate in the lower part of the recipient, while the lighter H2O molecules will be the first to evaporate. Such a process is called Rayleigh fractionation, and the same principle is used to synthesize heavy water. So we just, in fact, evaporate water and evaporate and evaporate and evaporate until in the residue the deuterated water is going to concentrate for the purposes that we need the heavy water, nuclear reactors, whatever. So, as I was saying, all water is not equal. While when the heavy water was discovered and deuterium was discovered, immediately so the discovery was made by Harold Urey in 1932, and immediately the effects of this water began to be studied. So the investigations were extensive, and we have observed that while the small or normal amounts, around 145, 150 parts per million, are not toxic, increased amount of deuterium exceeding 15% determine structural, metabolic, and functional alterations in various degrees. And this is due to many factors on which I'm not going to, so I'm going to present them, but I'm not going to insist very much. It has a lower diffusion coefficient, it enters more difficultly in the cells, produces the reduction of the substrate diffusibility through membranes, and reduce the speed of the cellular enzymatic reactions. From here derives the multitude of its inhibitory effects, including the vasorelaxing ones, which were the first that I have studied while I was only a PhD student. One of the unstudied byproduct of the separation process for obtaining heavy water is the so-called deuterium-depleted water. So, after this mechanism of distillation, we obtain some water that has lost a few molecules of deuterated water. And people have tried to continuously to increase that loss until they have obtained various concentration of deuterium depleted water. Compared to the biological changes determined by the excess deuterium in the water, at the level of the years 2015, there were about 25,000 papers only in PubMed, but probably there are much more papers because PubMed is not all-encompassing. The effects of reducing the concentrations are much less studied. There is only a small amount of data. So we have seen the effects of tumors transplanted in mice, anti-neoplastic, of course, of Dr. Schomley and other colleagues of his, anti-aging, cell stimulation, and radiation protecting effects. So there are many things that can be done with this water, but they are not absolutely and extremely studied. I was very happy to see that huge collectives with extremely wealthy, let's say, resources dedicate their time and effort to study the effects of deuterium depleted water. It is clear that a decrease of the deuterium concentration in the cellular microenvironment should produce also a decrease of the intracellular deuterium, which might interfere with the regulation of the intracellular pH, a parameter which is universally accepted to be a key one in the signalization of the steps of the cell cycle, thus inducing either increased, either reduced proliferation of the cells. The cells seem to be able to regulate the internal ratio between deuterium and proton, thus being able to coordinate and synchronize the proliferative processes. And using the deuterium depleted water, we might be able to alter or change these regulation system. The reconstitution of the aquatic environment with heavy water, with deuterium, light water, induces the stimulation of the growth and development of aquatic animals in a highly significant manner. And here I have surmised the idea that aquaporins, which are the water channels, membrane proteins which allow the water passage through the lipid bilayer of the cell membrane, while subjected to deuterium, have a permeability with at least 50% lower for heavy water, in the condition in which the environment water exists already a load of 150 parts per million deuterium. Water channels have been demonstrated to be able to let deuterium oxide pass, but much slower than normal water. So we see here the loss of permeability when we increase the amount of deuterium. Of course, the chemical and physical differences between heavy water and normal water are summarized in the next table, and we can observe the major types of isotopic effects. Solvent effects, which are tied to the viscosity and the dielectric constant, primary kinetic effects and secondary kinetic effects. And those reflect the involvement of the protons in the chemical reactions, or deuterons. Primary effects appear when the proton is directly involved in at least one essential step of a chemical reaction, like the protonation or deprotonation. For example, the ionization of a base or an acid can take place 3 to 7 times slower in heavy water than in normal water. Secondary effects reflects the replacement of protons with deuterium in other sites, separated from the primary ones, these effects being less important, but also presenting a reduction in the speed of the reaction. So all those things that have been demonstrated show an alteration of currents through the proton channels at the replacement of water with deuterium on one side or the other of the cell membrane. Those patch clamp studies are very good and very important in studying those effects. Beside the effect of reducing the current, deuteration slows channel opening at least 3 times. So an extra channel permeability, the so-called leak currents, are also reduced until almost zero. The isotopic effects on the ion channels could be summarized like this. Deuterium passes through the proton channels, but the conductance for deuterium is about half. The activation of the voltage-dependent channel is 3 times slower in heavy water than in normal water, and unlike protons, deuterium does not leak through the lipid membrane. You're going to say, you're talking a lot about deuterium, but we have a very small amount of deuterium, 150 parts per million. As Professor Boros demonstrated yesterday, we have lots and lots of protons produced, something to the power 10 to 19th or something like this. So 19-0. So it means if we 29, not 19. So even much more. So it means for a Google of protons produced, we are going to have also a Google of deuterons, not produced but involved in this reaction. Another nice analogy was made by Professor Zubarev, who said, we don't know if it's sand in the gear or oil in the gear. I suppose it is sand. It's much more common sense. It might be also oil, but we don't know for sure. At least I don't know for sure. But if deuterium is sand in the gear, then removing at least a certain part of this sand is going to improve the gear, is going to improve the yield of all kinds of biological mechanisms. Thus, it will improve the activity of the cells. Thus, it will improve the activity, the reactivity of the whole organism. Quite important, we have to remember that neoplastic cells are not normal cells. So probably because those important differences between neoplastic cells and normal cells, neoplastic cells are not stimulated but are perhaps even inhibited in by the administration of deuterium-depleted water, as Dr. Schomer already demonstrated. A series of hypotheses state that water organizes itself in a series of supermolecules connected by hydrogen bonds. It is the so-called hydronium ion with the first hydration envelope, which then evolves towards the secondary envelope of the hydronium, 9H9O4 and so on. Quite nice, quite interesting formations, which for me were surprises. And then when I read about them, I understood why these supramolecular organization explains several especially difficult to grasp characteristics of liquid water. The so-called Grotius effect, the abnormally strong ion conductivity of water, the translocation phenomena to water channels like the gramicidin channel. Gramicidin is an antibiotic which is used to block that channel. So, we can see here which are the hypotheses of the proton transfers through proton channels throughout the membrane. It is the model of the proton wire of Nagel, which says that the hydrogen channel is not a pore field of water, rather a channel made of lateral residues amino acids in water molecules. And through this construct, the protons pass either using a Grotius mechanism, either by a proton wire, which necessitates proton leaps. Here we have the proton hopping Grotius mechanism, which says that water goes from a pipe to another one, and the other one is the vehicle mechanism, which says that unlike the protons which push one another like a string of beads, here the whole molecule of water passes through the channel. We don't know exactly. But of course, due to its superior mass, deuterium is not able to make such leaps, because it does not allow for the formation of the superior ions of hydronium, which will not allow a complete Grotius effect. From here comes the sluggishness of the channels and ion currents induced by deuterium. So the premise of the theory is that in the case of the replacement of the deuterium present in normal amounts of water with hydrogen, the incubation or immersion of the organisms, cells and so on, in environments reconstituted with deuterium-depleted water will activate the proton transport from and within the cell accordingly. Of course, we cannot postulate a complete reversal of the heavy water-like effects by the simple replacement of regular water. It might be possible that the 6 to 7 time-fold reduction of the deuterium concentration outside the cell, compared to the 145, 150 parts per million normally present in the water, might have an accelerating effect on the same phenomena which are slowed in the case of deuteration. Diminishing the deuterium concentration will produce the activation of the proton transport to the membrane, will produce an increased efflux of proton towards the outside of the cell. Now I realized that in fact they go from the mitochondria in the cytosol and then from the cytosol outside. I was not thinking about mitochondria because I'm a physiologist and my knowledge stops at the level of the membrane. Sub-membrane mechanisms are not my forte. And of course it will produce consecutive changes of the intra and or extracellular pH. And deuterium has a long effect also of microtubule stabilization and the reduction of their synthesis. So if we eliminate deuterium, it means that the microtubule are going to work much faster and much easier, and we know that microtubules form the, let's say, railway systems inside the cell, allowing division, allowing vesicular traffic, allowing the movement of synthesis vesicles within the cell, thus increasing metabolism and of course increasing proliferation, increasing division of the cell. A special feature of the voltage-dependent channels is the sensibility to the internal proton concentration, both to the pH O and so outer and internal pH. The variation between those values, both internal and external, can change water flows. Membrane proteins can be protonated at sites that can be accessed either from the internal side, either from the external one, but not simultaneously, not in the same time. External protonation stabilizes the closed channel, while internal protonation stabilizes the open channel. So, what happens? If we reduce external protonation, the channel will close and the traffic will reduce, while internal protonation is going to open the channel and allow the passage of various ions, thus inducing changes in the cellular excitability, because we know, according to the Goldman-Hodgkin-Katz equation, which is the basis on which we calculate the action and resting potential, that the amount of ions that move from one side to another of the cell is of paramount importance for the level of resting and action potential. In the same patch clamp study, De Corsi demonstrated the alteration of currents through proton channels by the replacement of water with deuterium. Beside the effects of reducing the current, deuterium slows channel opening at least 3 times. And of course, I repeat, the so-called leak currents, which are important because it allows to rectify the potentials. The leak currents are going to be reduced until almost 0. Diminishing the deuterium concentration will activate the proton transport. Will produce probably an increased efflux of protons towards the outside of the cell. If we remove protons, then we produce a consecutive decrease of intracellular pH. Intracellular pH is one of the triggers of cell division. We know that the sodium proton antiport is very important in the detection of cytosolic pH. There are several systems that regulate cell proliferation, but among them is their sensitivity to intracellular pH. Those mechanisms are clear in a series of plants, in inferior animals, and isolated cells. In mammals, this mitogenic stimulus is necessary but not sufficient for inducing cell proliferation. The stimulating effect of the proliferation is perceptible and reversible using blockers like amyloid that I tried in cell cultures, and it reduced the stimulating effect of deuterium-depleted water. And in a smaller degree, using potassium hydrogen pump blockers like lansoprazole. In vivo, this effect seemed to be perceptible only in tissues with fast growth and proliferation rates, like the hematopoietic or epithelial tissue. And it is not found in neoplastic cells, which have as proliferation inductors, the oncogenes, and various other external growth factors. And of course, what we call cancerogenic substances and tumor-inducing agents. Another membrane component might be the potassium proton ATPase. The knowledge concerning this membrane pump is less detailed than from other proton transporters. However, there are specific inhibitors, derivatives of omeprazole, which are potassium dependent, potassium proton dependent. So, for those who have gastric problems, they know that omeprazole, lansoprazole, pantoprazole, and whatever other drugs like controlog, for example, are used for blocking the secretion of protons in the stomach, in the gastric juice, in order to reduce the hyperacidity and so on and so on. But the same pump is found throughout the whole organism, and we can use it, of course, in cultures, in cell cultures, or we can use it in vivo experiments. The increase of the proliferation speed of cells produces the so-called overall hyperplasia. Neoplastic tissues may be adversely affected by these mitogenic stimuli because they usually lose their control points in the mitogenic process. Thus, the beneficial effects of deuterium-depleted water described by several authors might be due to the fact that the efficiency of their mitosis is reduced due to the increase in the normal pro-mitotic factors. So we know that cancer grows in a wild manner due to its own process and to its own lack of control. What if we could increase a little this control? It might reduce the proliferation of cancer cells, thus reducing perhaps the risk of metastasis, thus reducing perhaps the risk of recurrence, thus improving the overall survival, thus helping various, let's say, traditional therapies like radiotherapy or chemotherapy, and helping them to reach their point by reducing the viability of the cancer cells. Who knows? And also, a very important thing that has been demonstrated, and for which, together with other colleagues of mine, we have received a gold medal in the Inventix Salon in Geneva in 2003 or 4, something like this, is the immune stimulation. We have demonstrated in rats, of course, that feeding them increased the immune parameters, both cellular and humoral immunity. And we know that immunity is important, and usually cancer patients are in various degrees of immune suppression. Maybe because of the cancer, or maybe because of the treatment. However, such an immune stimulation might improve the defense against neoplastic cells in metastasis, or perhaps maybe even in primary tumors. As you have already seen yesterday, those are ATPases which push the protons. Those very elegant motors that we have seen yesterday in motion. I did not have the time to find a nice movie for those. But you see, they have rotors, they have stators, they move and they push protons in various directions. Quite interesting. There are two types, F-ATPase and V-ATPase. So there are V-type proton pumps, most common the sodium hydrogen exchanger isoform 1, and they have them in the plasma lemma, the osteoclast, macrophages, tubular cells of the kidney. Proton movement through these pumps is proven by the inhibition of the hydrogen traffic by the macrolides bafilomycin and concanamycin. Those are antibiotics which make their action directly by blocking those pumps and, in fact, poisoning bacteria to death. But if we give them in small amounts, we can reduce only the activity of the pump without killing the cell itself. And also, mammal cells, for example, are much more resistant than bacterial cells to those types of substances. It has been demonstrated that its activation of the sodium antiport that we have demonstrated here increase outflow of protons, and produce intracellular alkalinization. There are several reports that associate intracellular pH with apoptosis and proliferation. Among these is intracellular acidification with apoptosis. Growth factors induce a lot of signalization cascade, inducing DNA replication and cell proliferation, among which of capital importance is the stimulation of ion transfer to the membrane. The most significant is the amyloid-sensitive sodium proton exchange, which have as effect the intracellular alkalization, induces cellular hyperexcitability, stimulates the synthesis of growth factors and other bioactive factors. The sheer alkalinization of the intracellular environment might be enough for inducing proliferation. So, all those effects, a reduction of those effects might inhibit the development of tumoral cells. And also, just a few words about the last results that have been published. Even if neurons, for example, are cells which do not divide, and their metabolism is quite slow from the point of view of the proliferation factors, they are sensitive to this deuterium depletion. We have demonstrated using specific behavioral tests in rats that deuterium depleted water has stimulating effects on long-term memory. We made mazes, we made various types of evaluations and so on, together with my colleague here present and some PhD students. And also it has anxiolytic effects. So it means it's quite important. There are the so-called mazes, yeah? Y maze, elevated plus maze. We put the rat on a bench, something like this, and of course he's going to be afraid to jump because he does not know, he cannot see what is on the other side. But a normal rat finally is going to find its exploratory behavior, and he's going to start searching until he's going to jump, or he's going to move in the various arms of the maze, even if he does not know what is this, because rats are very curious, like any other animals. However, if he is an anxious animal, he's going to start checking, and he's going to be paralyzed. And of course, we can reduce those behaviors by either giving him anxiolytic drugs as controls, or by giving him deuterium-depleted water. Just a few slides and I finish. The presumed mechanism, we don't know. Improvement of the redox status of the brain cells. Here we need biochemists and molecular biologists to study these. It's very difficult, it's very complicated. And we know, we have identified since a long time that deuterium-depleted water in solution has vasodilatory effects. So, for example, it might increase the brain oxygenation by increasing the blood flow to the brain, thus increasing the activity of the brain and improving the memory and reducing the fear. Of course, we have to do many other things. We have to make experiments with immunosuppressors and tumors. We have to make measurements of the transmembrane potentials and ion currents. We have to use selective channel blockers. We have to use also bacterial development because they have serious protection mechanism against external and internal pH changes. So there are many things that can be done. And of course, everyone has lots of ideas. Perhaps we can do them, perhaps we cannot. Just a few words about our history. I wanted to make...
How long? You have
2 slides.
Two slides.
Remember, I started 20 minutes later.
No. I mean,
Just add it. Okay, thank you. We have, only in Romania, they have published 15 scientific reports, 39 presentations, 23 presentations, internal conferences, 9 patents, and more than 25 World Journal publications and 11 grand prizes, and gold medals, and so on. We have all those patents for installations and procedures and so on. Prizes, titles, many things. Because I have been asked several times, what is your interest in deuterium-depleted water? So if we produce it, then we should study it too. Thank you very much.